Major questions. One of the major questions is when do we see, or to say, when did that last common ancestor exist, and what is the context in terms of the geological record as well as the biological history of the planet in terms of when do you have a biosphere? The origin of life is not the same as the diversification history of life. It's not the same as the origin of a robust biosphere that could persist over geological timescales. But to get at those questions, we have to at least be able to put one stake, one spike in this record, and that important spike is the timing of the coalescence of all extant lineages on Earth today, which would be LUCA. So when I say LUCA, or the last universal common ancestor, this is not the first cell, this is not the first life, this is not the earliest living thing. Um, there's nothing necessarily special about LUCA. You always get a last common ancestor for any group following a pattern of speciation and extinction. And so LUCA just represents the earliest point that we can work back to, which gives a younger bound constraint on all of the traits and characteristics we observe at the time of LUCA, which would be cells that share a lot of the universally conserved molecular machinery that we see across life today. Sorry, I have my co-presenter here. He might be jumping in and out of the screen. Okay. So to illustrate what I mean by a last common ancestor and how what kind of inferences we can make with cladistics, an example I like to use is the evolutionary history of mammals. So here we have a phylogeny or a time calibrated using the fossil record phylogeny of the evolution of amniotes. Um, so we can see we have sometime in the late Carboniferous, the last common ancestor of all amniotes, um, and then a major divergence between reptilians and mammalian lineages, or synapsids and sauropsids. And then we see the reptilians continue to diversify to turtles, lizards, archosaurs, and all of their extinct relatives, which are not drawn on this side of the tree, and birds, of course, which are a branch of archosaurs. But on the mammalian side, we do see the drawn these extinct ancestor stem groups. So we have pelycosaurs, therapsids, cynodonts, close relatives of true mammals, mammaliforms, and then all the way up here in the mid-Jurassic, we have the last common ancestor of all extant mammals, the true mammals, including egg-laying mammals like echidna and platypus, and then therian mammals, including the two major groups extant today, marsupials and other placental mammals. So we can see there's a complex evolutionary history between the divergence of the mammalian lineage from other reptiles and the crown group of all mammals that have all shared derived traits that we associate with mammals today. And so this last common ancestor, this coalescent, this is like the LUCA of mammals. However, we can see there's a huge difference in time between this common ancestor and the origin of the mammalian lineage that reaches back across all of this stem diversity to the divergence from their amniote. So this is like, 150 million years or more time than crown group mammals have been around. And if we didn't have any of these fossil groups that document the morphological and physiological changes that were acquired over this huge interval of time that separate mammals from other amniotes, you could imagine the difficulty we would have in trying to assemble this stepwise evolutionary history. And with microbes, that's always the problem. We barely have a fossil record, and so inferring all of these deep ancestral traits and transitions is extremely challenging using only molecular sequence data. So when we're talking about LUCA, we are talking about in terms of instead of just looking at a tree of amniotes, a tree of say the total history of cellular life on Earth, LUCA is basically this node, crown group life, life that has all of the shared derived traits that we see across diversity today. It is not the first cell, which would say be down here, and is not particularly special. If you went back in time to the Jurassic, when crown group mammals were around, there were also still some stem group mammal groups like mammaliforms hanging around. And so if you were to do molecular phylogenetics not then, you would infer, that a crown group of extant, whatever you would define as mammals, would actually be quite older. So the fact that the crown group is a certain age is a factor of the extinction process of more deeply related groups. Just like if there were more deeply branching cellular lineages before LUCA, and if they had survived, LUCA would simply be redefined earlier in the tree. So taking this framework and once again going back to very early Earth evolution or early life evolution, in a really simplified tree, we have bacteria and archaea, eukaryotes grouped somewhere with archaea, so we can leave them out for simplification. Tracing back to a last common ancestor of LUCA, and then there's a stem lineage leading to LUCA, and somewhere along this lineage, we have the origin of life, prebiotic chemistry, translation, DNA, RNA, the first cells, ATP synthesis, all of that shared machinery evolves somewhere here. And this is incredibly frustrating because this is in a way, this is like where the most interesting evolutionary events occurred, but it's very hard to interrogate uh, these ancestral states. So very early on in prebiotic chemistry, we can simulate this in the lab, or simulate, we can actually do prebiotic chemical experiments. We can test the plausibility of different organic synthesis of amino acids or RNA or ribose or whatever in those conditions. So we say this is bottom up and deterministic to a certain extent. Meanwhile, on the right side, we can work backwards. This is top down. This is historical. We have an evolutionary record preserved within genomes. But in between, I like to call this dark age. It's still historical, but it predates coalescence of the genetic record. There's very little, if any, genomic information to lay out exactly how you go from an origin of life or prebiotic chemistry to cells and to this common ancestor. So trying to push this record back further is an important goal of comparative genetics. And one of the most important parts of that is estimating the time interval between origins and LUCA. So, as I mentioned earlier, this is made all the more challenging by the fact that our earliest geological record of life only shows up, well into the Archean. Um, and so the age constraints that we can draw from this record are largely not very informative. So the earliest, really uncontroversial evidence we have of a biosphere is stromatolites in Western Australia dating to almost 3.5 billion years ago. And these are almost certainly bacterial in origin, although their precise taxonomy and physiology is unclear. And around that similar time, we see other evidence of biological material, microbial mass that have been preserved. So really, this record only shows up at about 3.4, 3.5 billion years ago. That means that we have about a billion years of uncertainty between the moon forming impact and the first unambiguous evidence of a microbial biosphere. And somewhere in that billion years is the last common ancestor of all life. And really, the only tool for narrowing it down is molecular phylogenetics, molecular clocks, estimating divergence times based on sequence data and evolutionary models. And this is very hard to do. And in this talk, I largely want to go into why this is an incredibly difficult problem and talk about some of the headway that we think we've made in addressing it. So before I do that, a little bit of a background on molecular clocks. This is not a methodology talk, but hopefully I can give you enough of the moving parts that it'll be intuitive as to how we are trying to improve these methods. So essentially, a molecular clock is estimating divergence times of groups in a sequence, a phylogenetic tree generated from sequence data by measuring changes in sequence under some evolutionary branch rate model, constrained by external absolute age constraints. So a fossil record or potentially geochemical data. So to do this, we really need four things. We need a rooted species tree or phylogeny based on sequences. Typically, we use protein sequences because they're more conserved over longer timescales. Um, so to do this, we have to find sequences of proteins that reach back far enough in time that have reliable phylogenetic signals. We have to align them to be able to extract the information of the state changes in a data matrix that we use to generate a tree. There are branch lengths on that tree that represent evolutionary distances. And converting those distances into time is really the goal of the clock. We have to have priors on the age of the overall tree to start from. And really, this is going to, you can think of the prior as an age range that integrates over all the reasonable hypotheses that we're trying to test with the sequence data, which would then generate our posterior age estimate. We need calibrations. We need some internal constraint in the tree for how old different groups are, and those can be older bounds or younger bounds using various sources of evidence or logic. How you impose those calibrations is a very complicated topic, but typically the complexity arises from data sets that have a rich fossil record, which can shape the priors that you put on these nodes. Because we have so little information within this deep microbial tree that's potentially informative in terms of calibrations, we can actually skirt a lot of that complexity, but that also leaves much larger uncertainties. And then finally, some kind of a model, an evolutionary branch rate model. So we're going to be drawing relative rates to fit onto branches from distributions, and these distributions can have different shapes. You can apply uh, knowledge that we have about how groups typically evolve. For example, an autocorrelated branch rate model implies or takes advantage of the fact that more closely related groups in the tree of life tend to evolve at similar rates. And so you can have parameters that determine local rates in the tree so you're not overly penalizing seeing, say, like a high evolutionary rate independently on many branches. There are also tree shape parameters. For example, if you imagine a neutral evolutionary process of extinction and speciation, that changes the expected distribution of nodes in time across the tree, where more deeply branching groups will be represented by more and more sparsely distributed nodes. So that would be, say, a birth-death model. Testing these models is another kind of really complicated thing that we're not going to get into today. But this is an essential component of any kind of molecular clock and where a lot of the uncertainty, it turns out, is derived from. So how this works in practice is we have a lot of inputs going on here. You have an input of the tree topology and the sequence data. Um, and then you have all of these priors and values that you can set or they can be fit by your model. And really what you're doing in a Bayesian molecular clock analysis is you are determining what is the likelihood of the observed sequence alignment data given a tree, given these constraints, and given the rate model. And then you're sampling a lot of solutions. You're going to be sampling a lot of solutions for your evolutionary rate model to generate an ultrametric tree, a tree where all of the tips are at time zero, because we know that that's just the ground truth because we sampled the DNA at time zero. But there are many possible fits that have similar likelihood. And so then you sample across these, and then you generate a probability distribution for the ages of all these internal nodes in the tree, given all of these conditions. So in estimating the ages of groups, the fossil record is clearly extremely critical. And we run into problems, though, as we look earlier and earlier into the ages of particular groups, because it's expected that a group on the tree is older than its fossil record. And the further back in time you go, based on the probability of actually finding fossil evidence, and the number of traits that are um, specific to a particular group in the tree that could be even possibly recognized to define the appearance of a group or the FAD first appearance datum, you can see that it's expected very early on in the divergence of a clade that you would not see any evidence of it. And this is just to illustrate how really for constraining these ages, we're talking about conservative younger bounds. So for example, if I have the stromatolites, the oldest stromatolites are 3.46 billion years old. That doesn't mean that's the time of the first bacterial life. That's just a younger bound. That means that I would expect that the diversification of bacteria must have been at any point earlier than 3.46. That would only be a one-sided constraint in in my age estimate. If I had hundreds of examples of these stromatolites distributed across time and geographically, and then and then you saw the first one being present at, say, 3.46. If you had a huge sample size, you could infer this first fossilizable apomorphy, how likely it would be that there would be a potential future find given the distribution of existing finds, but we don't have anywhere near the data set in order to estimate an older bound for groups based on the fossil record. So a lot of like the power of fossil calibration that we would see for, say, if we were looking at the evolution of early tetrapods or the evolution of birds is just not available for these analyses. So we are going to be using a simplistic, very conservative set of older and younger bounds for these deep time divergences. So several efforts have been made up until now to try to date LUCA using these approaches. I'll very briefly go over, really that recent history and what these ages are and how I think we can do a little better. Um, so, for example, here's work in 2018. Um, to having a big rooted species tree across the tree of life, including eukaryotes and origins of mitochondria and plastid, archaea, bacteria. So we have the LBCA, the last bacterial common ancestor, and the LACA, the last archaeal common ancestor, and then working further back to LUCA. So this approach used calibrations derived from different groups of archaea derived from the eukaryotic fossil record and cross bracing. So you know that like the mitochondrial ancestor, which is bacterial coming into eukaryotes, has some internal relative age constraints between the alpha proteobacteria that it's derived from and the eukaryotes that it invaded. And the same with plastids, cross-linking cyanobacterial and in plant evolution. So when this was all applied, you can see that the resulting age was quite variable, but also quite old, potentially pushing, all the way back to like 4.2, 4.3 billion years ago. But you'll also notice something, which is uh, these ages for archaea and bacteria are nearly identical to each other. And also, we can see that the ages within the archaea and bacteria well, especially in the bacteria, tend to be pushing up against these bounds. So you can see the last bacterial common ancestor um, is estimated to be only a little older than 3 billion, potentially as old as as 3.5 billion, which would imply that those stromatolites are stem bacteria, that they're actually not in the crown group of bacteria. Um, so groups seem to be pushing a little young in the tree. Um, and also there are older bounds that could potentially be, I think, a soft older bound in this point for this analysis of looking at late heavy bombardment involving some kind of impact frustration. So more recently, we have a phylogenetic analysis or molecular clock analysis that used much more sophisticated models and a bigger data set and concatenated ancient proteins that are involved, that duplicated before LUCA. So we have like pre-LUCA branches that can help push ages to be more reasonable potentially. Um, and this paper got a lot of attention, and it resulted in an age, most likely age estimate for LUCA of about 4.2 billion, with a very precise age estimate of a 95% confidence interval between 4.09 and 4.33 billion years ago. This is very old and very precise. So for me, this was a bit concerning because typically for really, really old ages, when you see that much precision and they're that close to the older bound implied in the model, what you're seeing is the probability distribution pushing up against that wall where it can't go older, but the data wants to push it as old as it can go. And to me, that suggests that these results potentially um, were biased in some way. So when you see your age estimates pushing up against an older bound, a very, very active calibration, as we would say, that's a cause for concern, and it means we need to do some testing. Um, so this is what often can happen when you see very divergent groups that are present on long branches. That's because a very long branch in a tree means there's a lot of sequence divergence along that branch. This can be this can mean two things. It can mean, first of all, that there's simply a long interval of time at the expected evolutionary rate, or it could mean that there's a short interval of time at a very high rate, so a lot of change accumulated. The problem is that molecular clock rate models favor or give a higher likelihood to rates that are uniform across the tree. And so it's always going to favor inferring a very large interval of time rather than a very high rate for a particular branch. And so that means for a very old branch that's very, very long, the model is going to be biased toward imposing a very high, sorry, as large of an interval of time as possible, which is going to push up against the older bound and push recently against younger bounds. So a good analogy for these problems, and certainly helpful in talking with planetary scientists and geochemists, is to think about the problems with trying to date planetary events on the early Earth using the very oldest zircons. And I'm going to use two particular types of analogy to explain the processes that we're trying to manage here. So the first is alteration. So if you have a chemically altered zircon, it can be very hard to reliably date it or infer anything from its history, and these would typically, be discarded in favor of ones that have experienced less alteration. Although if you're trying to find the very oldest stuff, you have no choice but to use, altered, damaged crystals. So just like a zircon, ancient protein sequences can also experience alteration. So they can be recombined. They can be evolving at very high evolutionary rates that overprint and erase the phylogenetic signal and replace it with other bias signals. And these typically lead to an overestimation of the age of LUCA. So the opposite with the zircon, that would lead to an underestimation typically in its age. So we could say that these kind of sequence alterations, in addition to the expected underlying evolutionary processes, will bias toward having high evolutionary rates and older ages. Similarly, these very oldest zircons are not in host rock typically that's as old as the zircon. They're detrital. They've been recycled several times. And it turns out that ancient protein families can undergo a very similar process, um, uh, basically through horizontal gene transfer. So you can imagine very early in the history of life, ancient protein genes are going to be transferred between lineages just like they are today. And some of these lineages may actually predate LUCA. And so if you were to then look at those groups and infer that you are fairly estimating age of LUCA, you may actually be estimating an even earlier split in the tree because that genetic material did not coalesce in LUCA, but to an earlier point. So these detrital transfers would result in an overestimate in the age of LUCA. So to show how alteration can affect these ages, I have an example that I did some analyses of for ATP synthase proteins. So these are very well studied proteins. They are involved in synthesizing ATP. They make the catalytic and non-catalytic domains of the six-cylinder rotary motor that generates ATP from a proton motive force. Very early on, these were used in estimates for rooting the tree of life because these ancient duplications mean that these sequences can be used as one another's outgroup. And we see in just a phylogenetic tree that I generated using a sampling across archaea and bacteria. So we have, because this ancient gene duplication before LUCA, and then we have the non-catalytic subunits that trace back to LUCA. The red ones are archaeal, the blue ones are bacterial, and then the same thing with the catalytic subunit. So this is really nice if you're trying to reconstruct the topology of the tree of life. However, you'll notice these branch lengths are huge. The evolutionary distances diverging, the evolutionary distances following LUCA suggests that there was a lot of selection um, and diversification between these two groups. So, like for example, with this non-catalytic F-type synthetase, there's almost been 10 times as many substitutions, almost total saturation along this branch in the stem as there has been in the crown group. And this was one of the main gene families that was used in the paper I just described for dating LUCA. So right away you can see what the problem might be here. So if I apply a very simple molecular clock to this, so at uncorrelated rates, no birth death prior, and I just give a very conservative calibration that the archaeal and bacterial crown groups are between 4 and 3.5 billion, and I have a normally distributed root prior at around 4 billion, so that gives a range with a standard deviation of about, you know, 0.2 billion. So really, my root is estimating potential ages of LUCA between 3.5 and 4.5 with a hard bound at 4.5. I get the ages that you would expect. I get these uh, these crown group ages for the domains are pushing right up against that 3.5 range, and then the older bounds are pushing up against this 4.5. And if we actually look at the probability distributions, you can see, yes, they're skewed. So you can see the probability density, it wants to go older than 4.5 because those very long branches, but it can't. And so you see this pushing of all the probability density against the bound, which is likely increasing, artifactually increasing the precision for those age estimates. And I can test this because what if I just get rid of this older bound for the age of the Earth and I just let it go as old as it wants, as old as the data and the model are trying to push? So if I do that, if I relax the root prior to 7 billion and give it a generous variance, and I recalculate my posteriors, then yeah, the ATP synthases go to a mean age of between 6 and 7 billion years old, potentially even as old as like 7.5 or 8 billion. None of the probability density is younger than the age of the Earth. So the resulting age, so basically this shows that an ATP synthase phylogeny is useless for dating LUCA. It's been so altered by or deep branching substitutions between the different domains that the rate information is completely useless for inferring a reasonable age estimate for LUCA. So how do we overcome this problem? And I think the problem is how we're selecting data sets. Protein families for estimating LUCA are typically selected for being suitable for concatenation, which means congruence. It means that isolating or identifying proteins that have not undergone a lot of gene transfers, because you need to assume in order to concatenate multiple data sets together into one big alignment to make one big tree and one big clock model, you have to assume that all the proteins have the same history. Um, so the problem is gene transfers are more likely to are less likely to occur if or orthologous displacements of gene transfer are much less likely to occur if evolutionary branches are very large. And so by selecting LUCA or pre-LUCA paralog protein data sets that have not undergone transfer and so then are more likely to be congruent and so they can be concatenated, you are implicitly selecting for proteins that are highly divergent in their deep branches and are less likely to contain as informative rate information for dating LUCA. So these are exactly the proteins we should not use. We should be selecting data sets for proteins that show short branches separating archaea and bacteria. And those short branches are much more likely to reflect uniform evolutionary rates in these protein families across both the stem and crown groups. So, and this is because our null hypothesis is constant evolutionary rates. Very few processes will decrease a branch-specific rate of evolution in a tree. So if you see a protein with a very, very short internal branch separating archaea from bacteria, it is more likely to be reflecting a null model of evolution that is better fit by a molecular clock, with the exception of possible gene transfers that could be occurring after the divergence and swapping out back and forth. But I mean, at least substituting one problem with another doesn't run into an extreme overestimate or bias for the age of those groups. And finally, um, but this means we're going to find data for protein families that likely have a lot of gene transfers between archaea and bacteria. So I posit here, like basically, who cares? Don't concatenate them, and instead use individual protein families for individual age estimates for LUCA and have a population of age estimates. Once again, just like you would do for dating a layer of rock using zircons. Don't do one bulk analysis grinding them all together. Look at each individual crystal that you select for its utility and then plot it and have a population of ages and then do some kind of regression to infer the most likely age of the system. So I think I have a data set in my preliminary analyses that is really good for this, and I'd like to show some of the preliminary results that we have based on this, given the time that I have left. So this is the amino acyl tRNA synthetase proteins, in particular, class 1 proteins. These are involved in binding tRNA and putting the right cognate amino acid on it. There are two major classes. The class 1s are much more conserved in sequence and structure, although highly divergent. But I use this more closely related group of methionine, valine, leucine, and isoleucine synthetases for this analysis. To do this required a lot of very careful curation of aligning and sequence analysis. I can't get into all of it here, but this is not a trivial task that can yet be readily automated. So it's kind of closer to paleontology than the bioinformatics. But in being very careful about curating the data sets, I identified a few particular evolutionary events that would have conflated an automated like pipeline analysis. So here's the structure of one of these. This is an isoleucine synthetase. Um, this is from Methanosarcina. And you can see there's this conserved core in red. And then there are other regions that maybe are only found within this particular family of synthetases. And then in orange, and then even some, like, for example, in yellow over here on the right is this is only found within archaeal sequences. So you can see it's like this, there's a layer. There are regions that are highly conserved that are suitable for generating phylogenies for very deep splits in the tree. And then there's like extra regions that come and go in evolution and are less stable. These are kind of the accreted, more younger parts that might be really important for estimating the ages of more recent divergences, but not the deepest ones. And so if you plot these gains and losses of region to the phylogeny, yeah, you can see that this history is quite complicated. These red regions in the core are present in the ancestor. And then you can see as methionine branches off, it acquires these groups in yellow. As the other group diverges, it acquires these groups in orange. Then we see later acquisitions in green of domain-specific regions, and these circles represent losses, secondary losses. So regions of structure in these proteins, sometimes whole domains, are being added and removed, which really challenges alignment and reconstruction. So in accounting for all of this and going through a long recursive history of different evolutionary models and trims and all of that stuff we don't need to get into, we end up with this final phylogeny with methionine synthetases being the most deeply branching group. Then we have leucine, valine, and isoleucine. So we have four potential independent age estimates for LUCA within this phylogeny, and all of these branches before the colors represent divergences before LUCA. So we can run a molecular clock on this big tree. But to do that, we have to account for some of these alterations I mentioned earlier. So it turns out that um, in an earlier analysis, I saw a very deep branch of methionine synthetase leading to Hades archaeota. And I was like, well, that doesn't make any sense. Why isn't it going with the others? And if you carefully look at the sequence, it's because half of the gene groups with bacteria and the other half of the gene groups with archaea. So there was some kind of a gene transfer and recombination so that within Hades archaeota, this is a chimeric gene. It violates one of the base assumptions of making a phylogeny. And so it was averaging the evolutionary signal between bacteria and archaea. There's no way for a molecular clock to treat this fairly, so we had to detect it and remove this sequence from the analysis. Nobody has yet done a consistent um, assay or a screen to identify all of these partial recombinations that could potentially be negatively impacting these analyses for ancient proteins. So certainly, um, as I say, once again, just like zircon, some regions of the protein can vary in their history and age. So you have to be careful about what part of the protein you use to do the analysis. Similarly, there are recombinations, big recombinations in different protein families. So if you look at the bacterial leucine tRNA synthetase, so all of these isoleucine, valine, and leucine synthetases have a big extra proofreading domain to make sure the right amino acid has been added. However, the bacterial leucine proofreading domain isn't in the same part of the protein. It appears to have been at some point removed and inserted about 50 amino acids downstream. And this almost, if you look at the three-dimensional crystal structures, this change is nearly indistinguishable because the loop that it was transferred to is only a few angstroms away from its original position. So depending on the type of structure-based alignment you're using, you not even be able to detect this. But this massively disrupts the alignment and needs to be accounted for in order to get fair evolutionary distances. So when I do this, and I apply the same constraints that I mentioned, that I use for the ATP synthases, we see a very different story. We see valine, methionine, and isoleucine synthetases, the LUCA estimates for each one of these, all being pretty similar to each other, and none of them push up against this older bound. And so they are all consistent in arguing for a LUCA between 4.15 and 3.85 billion years old. So you'll notice that this is about twice the uncertainty as the previously published results. But while there are certainly still biases involved and assumptions involved here, these ages are not pushing up against the older bound, which suggests that there is not a long branch, deep bias producing false precision or an overestimate of age for these groups. But leucine's weird. It still doesn't push up against the older bound, but it's not consistent with the others. And it turns out this is a really interesting story that I just want to end on. So why could leucine, the LUCA node for leucine, be way older than the others when we get our estimate for ages? One possibility is that leucine synthetase is just evolving way faster for whatever reason. But if that's the case, we would imply, looking at the maximum likelihood tree, that they would have longer relative branch lengths. And so like the leucine group should have, a much larger tree height to the root than the others. But we don't see that. That's not what the phylogeny looks like at all. We could infer, once again, maybe the overall rate wasn't high, but maybe there were some deep stem evolutionary divergences. But once again, that would assume that the overall height for this group would be much greater in the maximum likelihood tree, and it's not what we see. We see this. The overall distances from the crowns to the root are similar. And so my hypothesis is this might be a detrital HDT. Because if we imagine there was some pre-LUCA cellular lineage that transferred its leucine tRNA synthetase into either the archaea or the bacteria, now the coalescent goes back here, not to the time of LUCA. And if you do that and draw your tree, you get a node for the common ancestor of archaeal and bacterial leucine synthetase that is much older. But what it also does, you'll notice, is it makes both these stems both get longer, not just one or the other. That single transfer means that both have a longer history back. And I don't know how else to get that symmetry in having both deep branches be similarly longer than what they would expect to be. So this might be some kind of relatively unique evidence of early pre-LUCA cellular lineages sharing genes with the early domain ancestors. But you can also see how it would bias toward overestimating the age of LUCA for that node. So moving forward, what I hope to do is apply this for dating hundreds of ancient proteins that trace back to LUCA, and then coming up with kind of like a Concordia plot that you would do for dating zircons, and then plot them with a constrained versus an unconstrained age, and to identify the population of ones like I have here for ATP synthases that are just so altered that they're not useful for dating. And then you can see the isoleucine, valine, and leucine ones with their mapped uncertainties are much more consistent and are much less affected by relaxing this older bound, suggesting that these are appropriate for dating. And if you could imagine creating a whole population of these across many protein families and identifying a subset that look like they have not been so altered that they're not useful for dating LUCA, and then integrating these into a most likely age estimate for the last common ancestor. And as I have drawn here, you could also infer that if there were transfers between stem and archaea groups, some of those could be as young as the younger bound for those particular domains. So there might be a subpopulation to identify those transfers as well. And I went over all of these points. But the last thing I will add to it is it may be that the relative ages we can infer here are more precise than the absolute ages. So across these analyses, it appears that LUCA is predating the archaeal and bacterial crown groups by about 300 million years. And so therefore that means that fossil calibrated and improved model ages for crown group archaea and crown group bacteria will directly likely propagate to improving the precision for our age estimate of LUCA. And that's also certainly needs to be a priority, not just focusing on the deepest branches, but increasing the precision for the age of the crown groups and then using that as a prior. And yeah, thank you. I hope I have time for a few questions.
Thank you very much, Craig, for the great talk and for sharing your work with us. I really, really enjoyed it, and I'm sure um, everyone else here did as well. Yeah, I think we have time for a few questions.